Good morning, everyone. So I welcome you to this morning lecture. So I heard you're on holiday actually, so thank you for coming here, and especially those who are online. So for this morning, what I would like to present is really a summary of how we started metabolomics at the University of Strathclyde. Okay, so it will be a 10-year journey that we established metabolomics research at Strathclyde. So you bear with me, this could take long 10 years. No, but I'm going to stick with the time, right? Okay, so basically we are part of a big consortium in Europe, right? So which means we're not doing the job on our own. So for example, biological testing is done at Strathclyde, but we also involve small medium enterprises in Europe. So one example is, for example, we have a close collaboration with Pharma that's somewhere in Norway, as well as here AXA somewhere in Italy. Genomics is very important for us. That's looking at the molecular biology of bacteria and fungi we're working with, and as well as we want to look at the biosynthetic genes, what are actually how things are being produced in the metabolites. Then we collaborate with University of Würzburg as well as people from University of, they're actually a company called Matis in Iceland. Industrial processing is very important because you want your research to be translated. If you don't want it to remain in the university, you want that to be down to the or want to get out for application purposes. So we have, for example, very good collaborations with SMEs within Scotland. So we've got Jensen, who does a lot of processing work. Okay, so that's very near our university, MBL, Marine Biopolymers. Our work is with seaweeds with them. And then we've got also these researchers in west side of Scotland. So that's for the SAMS. Okay. Right. Then we have also genetic metabolic engineering. I think we have that as well in the institute, right? And we also closely collaborate with people here in down south of UK. So those are people in England, particularly down in Cambridge. Then of course, natural products chemistry, that's our expertise, yeah. But we work also with people in University of Würzburg. Now I think the natural products group in University of Würzburg is now in field. And then finally for sampling, so we cannot do that ourselves, we need to actually collaborate with ecologists. And we usually get them from the Mediterranean region or the northern part of Scotland. So here, for example, we collaborate with Watt. Yeah, I think Watt has a very close collaboration with University of Terengganu, and as well as people from Greece. So here we've got the university in Greece, where we closely collaborate regarding our resources. Okay, so not only does we belong to a European consortium, we also collaborate internationally. So down here in Australia, we've got the group of Ronald Queen. I know he just retired, but we still continue our collaboration with RMRT. That's Royal Institute, Royal Melbourne Institute of Technology. Right? We've got UPM, I do close collaboration with Nurkhalida, who was my former student. And then we've got here, I can't remember all the names anyway. So we've got India, Egypt, right? Nigeria, we've got, Egypt's here, okay. And of course, in the UK, and then very close collaboration with groups in Brazil. Okay, and lately we've got collaboration with Expo, which we are starting to build as well. Okay, now why did we go into metabolomics? If you are a natural products researcher, then I think you've seen this too much the So we've got 4 scenarios here. Our main aim as a natural products research is we want to have new compounds, new bioactivity, which means get 100% novelty. So which means you could prepare for a big publication, yeah, and you could actually sell that to your industrial partners. But that's not always the case. We could go into the second scenario known compounds, mu bioactivity, 50% novelty. So it's still publishable, but don't get me wrong, industry also wants that, yeah, because they also always want that you've got bioactivity with your compounds. It could be an old compound like curcumin, yeah, if it has very good bioactivity, they will buy that, right? So another scenario is new compounds, no bioactivity, 50% novelty. If you're a chemist, that could be very interesting, especially if you found a new skeleton. So maybe still publishable, yeah, but now a lot of papers or journals are actually wanting to get always compounds with bioactivity. If you don't get any bioactivity, it's more difficult to publish. And of course, the industry, industrial partners would not like to have that compound, no matter how complex your structure is. Okay, a big no-no scenario is you've got no compounds, no bioactivity, then you don't have any novelty at all. So these are the two scenarios that we don't want to occur in the lab. Okay, and that's the reason we went into metabolomics approach in drug discovery. So we started going to this approach that was 2011, 2013. We call it concept development. Okay, right? You can see how you did it. Yeah. Okay. So because our publications are actually turning, so here we still have very few publications from the group by that time. Yeah, so one suppose that this is a typical isolation work, conventional method, but we're already using isolation. And then regarding the concept of putting metabolomics together, we put it as a chapter review, right? So how do we see metabolomics? So during that period, we'd like to embed existing databases into our analysis of our spectroscopy data sets. Okay? And we did start using a lot of multivariate analysis software to investigate patterns in our data as well. So we've got 4 basic applications. First is to determine novelty from our extracts. So this could be plant extracts or extracts. So we want to find the best strains and the best species, plant species to work with. Second is the selection of the optimal process, especially if you're working with microbes. You want to have the best fermentation procedure. So which means metabolomics, the use of multivariate analysis is very applicable to that. So it's basically integrating metabolomics data with the processing procedure. Right? Next is to investigate bioactivity. I think this is the most widely used application in our lab because we could actually compare the chemical profile and correlate it with the bioactivity of our extracts. And lastly, we work closely with the molecular biology group to integrate the chemical data or the chemical profile with the genomic data. Okay, so which means we could actually confirm the expression of the biosynthetic gene clusters and also further investigate the biosynthetic pathway. So which means we could actually increase the production of compounds when a biosynthetic gene cluster is not properly expressed. All right, now we go to 2014 to 2016. Here we started method validation. As you would see here, those marks in blue are actually, those are the ones that we are, that has something to do with the use of metabolomics approach. So during this year, we are starting to actually publish more on metabolomic profiling. So we are actually being confident with our methods that we were using in the lab. So when it comes to that, we have already established our so-called metabolomics workflow. So basically, we still start with mass spectrometry, but the most important point here, what we were the first group who were using actually the advantages of a switch mode method. So during that time, you would see most papers are only using positive mode. So we were wondering, how are they going to see phenolic compounds, which are easily ionized in the negative poles? Anthraquinones, for example, they are difficult to ionize in the positive pole. So that way, in case we started using switch for positive and negatives, then we could actually see the different metabolites being ionized in both poles, or particularly just in one pole. Yeah. So because this is untargeted metabolomics, you want to see as much metabolites as possible. Okay, after that, to be able to see whatever the peaks that were ionized in the mass spectrometry, we go into data processing, right? And here we use MZ-3. Actually, in 2013, okay, that was still MZ-2, okay? And good that they were partners with us. So they came to Strathclyde and started redeveloping MZ-2. And now we've got the right MZ-2. So they used the application of MZ-mining to natural products actually started in our lab. Yeah. Before MZ-mining was only used, for example, to do physiological data. Yeah, but not on natural products research. Right. Once that was developed, we go into replication. So we have a so-called in-house macro. So we embed different databases. It could be marine, lit, or it could be dictionary of natural products. But this time, I think most of the time we are using the DNP as our database. And then we go into multivariate analysis, and here we put out also NMR into our data sets, as well as our bioassay results. And once we've got our metabolites clearly targeted, then we start isolation. So it is actually a targeted isolation work using metabolomics. Okay, so we just do a first fractionation and then put it into our metabolomics and do the multivariate analysis, and if we are able to define our target during the first fractionation stage. We don't do bioassay guided. Yeah, where you need to test each of the fractions and sub-fractions. We don't do that because you just lose your pure compounds, and at the end of the day, you've got the wrong target. Okay, now 2017 to 2019, we start to define discriminatory metabolites in our bioactives. And this year, in 2017, just after Alida left the Institute, okay, we won an award. Okay, this was actually nominated from the German phytochemical. Sorry, with the German Phytochemical Society nominated the paper, and we won the most innovative 2017 paper. Okay, further on, okay, I will show you a sample paper from this year. Okay, so this is actually a work from one of my PhD students, and later became my postdoc, like Alida as well. For moving here, I think she was first my postdoc in one of the European projects. So this one, Ahmed was actually looking into blue pills from the UK. So it's a British species of blue pills, right? And it was interesting to chemical profile this because there were no works done in it. And it was actually quite bioactive as well with some of their assays. Okay, right. Looking at the chemical profile, we use here principal component analysis. We can see that there is a very good chemical diversity when it comes to the bulks in comparison to the aerial parts. The aerial parts you've got here, like a very similar chemical profile. Yeah. Another thing, if you look at here, I think everybody knows this, the loadings plot. It just tells you the chemistry of the individual metabolites distributed in these left quadrant. Yeah. And we have actually this molecular weights between 400 to 500,000. Now, because we are interested in what type of chemical diversity is the bulks offering, then we did a molecular networking and see whether they are only concentrated in the bulk but that wasn't not the case because if you look at the molecular network, okay, you can also see the distribution of these type of compounds in other plant parts. Yeah, but it seems that diversity is more pronounced or expressed in the balls. Then looking further in the balls, you can also see that this chemical diversity tends to change. Yeah. So for example, we've got higher diversity during the spring season and less diversity during the autumn. Yeah. And summer, it seems to produce some unique metabolites as well, but not as much during springtime. Okay. Right. Now, next is to do an OPLSDA, so that's a supervised method, just to define the discriminatory metabolites in each of the plant parts. Okay, and we can see here, again, on the right quadrant, we still see the leaf, scape, and the flower, and here's the ball. Yeah, so what we have here is the loading plot, and here is actually the spores plot. Yeah, so using these individual spots here, and we could say, okay, these are actually the discriminatory metabolites from each of these plant parts. For example, here, although they have almost the same chemical profile, using this method, this model, we are still able to separate them and define the metabolites for plant parts. Okay. What is also good to see in the ball, they have actually lower molecular weight compounds in comparison to the aerial parts. So you see here the aerial parts, it's all of the molecular weight are more than 1,000 Dalton. So we were thinking that maybe whenever we see the balls, they're being glycosylated. Okay, so this genus is actually known to produce trichopenes. Okay, so here it is. Yeah, because we are still actually interested in the activity. It seems that when we do the activity analysis, that the activity is actually concentrated on the flower parts. Yeah, we also have very good bioactivity on the others. Yeah, in terms of growth inhibition, these are actually they were testing it against antibiofilm as well, right? But they said the best activity and the inhibition of biofilm was actually found from the flower parts. And upon isolation and also prediction, we could see here examples of highly glycosylated triterpenoids, hence the huge molecular weight of more than 1,000 Daltons. However, these type of triterpenoids, which were the aglycones, they were not glycosylated, they were found to be inactive. Okay, and they were also proven after the isolation. Okay, so these type of compounds were not. Okay, so that was one project. Now let's go to 2020 to 2022. So we are also as well here papers coming up for using metabolomics approach, but this time we are trying to shift. Okay, we are still using metabolomics, but we are trying to find new application. And this time we are actually using concatenated data sets. So what do we mean by concatenated data sets? We are using two and see whether markers to come out with samples. Yeah, we have one here. I don't that's not from this year, where one of my ladies was working with honey. Just imagine you put honey in a bar, Mars cake, or particularly in a bar. What are the type of compounds of metabolites you will extract? Just sugar, exactly, and that was what you were saying. But then later on, when we started tested it, there was a big difference with 5 active acids. The Malaysian honey was active, the New Zealand honey was not active. Okay, so there's a different chemical profile. So she used that technique to be able to differentiate and find the low yielding metabolite to make the difference between the bioactivity. Okay, further that. Okay, so I mentioned about the 2 analytical platforms. Yes, mass spectrometry and NMR, basically, we just want to use the advantages of these methods. Okay, so mass spectrometry, we know you are able to find compounds as low as nanogram levels, which NMR would not be able to do. However, the good thing about NMR, it is not dependent on the ionization capability, okay, of a compound. So, for example, you've got the zeros, the triterpenoids, they're more difficult to ionize in soft ionization modes, right? But then you do see them as the major peak in your NMR. For flavonoids, okay, flavonoids, no matter how little it's that, it will give you a very good ionization in the mass spectrometry. But when you look at your NMR, they're very tiny in comparison to your zeros or fatty acids. And that's the reason we are using our data sets. Okay? So how do we do that technically? So basically we put them in one Excel sheet. Yeah, but now this one's also automated. We use Simca. Right? So it's easier to do it in Simca. Of course, if you don't have Simca, you could do it like this. It's also doable. So let's use, just put that formula into Excel, and then you are able to do the analysis. Okay. So here's one sample project. This is actually a postdoc from Brazil who was interested in looking into a plant called Rhizomema intermedia. So this was actually collected from the Brazilian Cerrado. Okay, and this plant material is known to the locals to be used again as an antiseptic, antimicrobial, anti-hemorrhagic, or anti-inflammatory. However, Anna was more interested on the anti-inflammatory properties. And according to her, it is the triterpenes that are responsible for the anti-inflammatory properties. Okay. However, like the same problem I told you about honey, okay, inspection of the MS and NMR data doesn't really give her that clear definition of the biomarker when it's actually used. Yeah. So basically the question she was trying to answer is, when is the optimum production of these triterpenoids, anti-inflammatory triterpenoids? So her hypothesis was dependent on the seasonality. Yeah? Everybody knows that. You're working with plants. That season would affect the production of certain metabolites. Maybe not so difficult here in Southeast Asia because we have 2 seasons, wet and dry. Yeah. Well, I was arguing with her as well because she comes from Brazil and said, I thought Brazil also has 2 seasons. But according to her, well, not really the same habits. Yeah. So according to her, Cerrados, yeah, they're still divided. Okay, so the objective is to target the seasonal biomarkers, okay, from unique perceptible constituents. In this case, she used also her So in this slide, I want to show you before and after concatenation. So here in the MS, you will see that there's actually an overlapping of the samples during spring and autumn. So if the samples are overlapping, then you will not be able to define the biomarkers. In the NMR, we've got another scenario. So here, however, here we've got overlapping of the summer and the autumn. So separation was only done for spring and winter. So still difficult to define the biomarkers. But when she concatenated the data, she had a better separation of the samples from the food differences. So from here, she can now extract the biomarkers, okay, and pinpoint and define them. So extracting out the data from the so-called fused data sets. So here is actually her summer, and here what we've got here is her the chemical shifts. Yeah. So this is the ppm. So then that were actually the defined chemical shifts for her samples coming from the summer. Okay. She was focusing on this summer here because she would like to know when is anti-inflammatory triterpenes being produced. And when she looked at the mass spec from the concatenated data, then this was actually representative of the glycosylated triterpenes. sorry, not glycosylated terpenes, but just terpenes. So she was looking at a molecular weight of 400 to 500,000. Okay, so this is a summary of her results. So for the summer region, there was a higher pentacyclic triterpene content, which were made up of lupin and oleanase structures, and that is represented by this green box here, right? And this one here, the yellow box, you could actually find the triglycosylated flavonoids, and they were only observed in Spain. You could also read out the relative abundance of these individual compounds by looking at the so-called whisper plots. Okay, so, or whisper formulas. Here, for example, we've got a large amount of 4,1,2,8. So then during that time, she was then able to find the biomarkers frequencies. Now, the question is, when is the highest, when is anti-inflammatory triterpenes produced at what conditions? So we need a higher temperature of between 25 to 36 degrees centigrade, solar radiation at 2,500, lower humidity of only 50 to 70%. However, 70% humidity is already relatively high as well. And then rainfall of only 0.10 and 0.15. I think that is quite significant. Yeah, because that is a low rainfall. Okay, now let's go to 2023 to the present 2025, and we continue to actually innovate our methods, and now we are going to co-cultures and visitors. And looking at our publications, we now actually increasing or almost 100% using metabolomics approach in this case. So these are only the publications from the group. Of course, I didn't include here any collaborative publications. Okay, so here, this work was done by Elizabeth. So Elizabeth is actually having her writer when I return. Quite excited about that. And this work is on co-cultures of antibiofilm compounds. Okay, or co-cultures of fungi with antibiofilm activity. So why co-cultures? So we believe using co-cultures, this would reduce the production of new metabolites, decrease the yields of respective target metabolites, and it would indirectly improve the biological activity of the extract. So I'll give you one example here. So we've got here two species, Fusarium and Alternaria. They were actually isolated from two different plants. So using a similar workflow, she put them together in one plate, okay? And we do see actually there is already an obvious competition between the two. Then she put them together in one class into extraction work and more replication studies and multivariate analysis to still define their target metabolites. So from the monocultures, I'm just taking you back to the monocultures before she did the co-cultures. You can see here, for example, a very nice antibiofilm activity for the incubation of Fusarium in rice at 50 minutes. However, for Alternaria, it was a different media. So the best media was found in potato at 30 days. Now, here are actually the defined metabolites for Fusarium. If you look at the molecular weights, they were quite diverse because it's covering the region of 200 to 900,000. Okay, so she was actually producing different types of compounds here. When you look at the replication results, they were actually naturally producing metabolites already known from the start. So we've got picabrine, reversine, and fluxipyrrole. So that is a type of terpene. Right. So these are the structures. So you can see here, those are just examples of the chemical diversity this particular Fusarium was following. So it's not a series of similar compounds. You've got here a peptide, anthraquinone, and a tartine. Okay. when we come into alternaria, it's more homogeneous, yeah? So we are only dealing between 200 to 400,000. So that's the alternaria. Looking at the replication results, so these are typical alternaria compounds. If you look at the structures, okay, they are almost related to each other, following the same biosynthetic pathway. So it's following the PKS polypeptide synthesis. So we've got here actually alpha-phenols, lactones, lactophenols, like that. So those were the type of compounds isolated. But there was nothing really new to it. They are not unusual for these fungi. Okay, so now Elizabeth had a meeting with me. It's like, oh, this is not really interesting. Can we put them together? Maybe we get new compounds, right? Okay, so she did that, and the activity really produced. So here you look at the activity of combination of fusarium with another fusarium, yeah. The viability really went down, yeah. And here as well, yeah, there's almost 100% inhibition of biofilm activity. Okay? She was no longer observing any growth, and that is a combination of fusarium, two fusarium species, and a combination of fusarium and alpha. So that is actually a big improvement. So Elizabeth was right. The co-culture would improve the bioactivity and may also produce new metabolites. So here's the multivariate analysis. We can see here that two species were actually offering better diversity. Okay, but this one in the middle, it seems that it's not really that diverse. It's producing the same metabolite, combination of metabolites from one certain species. Okay, so BG is actually a combination of fusarium and alternaria, and BM is a combination of two fusarium species. Okay, now this is the loading spot to be able to define what type of metabolites is actually being produced here between the two diverse co-cultures. Correct. So she looked at this combination, fusarium and alternaria. Okay. So fusarium we know was producing high molecular weight compounds, quite diverse. You've got the peptides, the anthraquinones, and lactoferrins. But in this case, we do not see them. It seems that the dominant species is the alternaria. Okay. However, there was still a production of new lactoferrinones. Right? So which means that, okay, I would like to do this and see what are those things that represent snow. And I think when she finished the project, there were more compounds that she isolated. Correct. In the case of the two fusariums, okay, so what do we notice here? We look at the table. None of these replicated compounds really represent the fusarium genes. Yeah, so they were actually being isolated or reported from other species, but not fusarium. And there was an increase of unknown. So this was very interesting, okay? Because first thing, okay, none of them are fusarium species, which means there is actually an increased probability that these compounds are also new. So she further worked on this, and I think she had a very good result on that, yeah? Which I'm still not able to present. Yeah, because she's still going to have her viper on Friday when I return. So finally, to summarize, so mixed cultures of fusarium with alternaria improved the antibiofilm activity at 100%. So there was a dominance of the biosynthetic gene for alternaria, and mixed cultures of two fusarium species also increased or improved the antibacterial antibiofilm activity with the increased production of metabolites. I would like to add here, increased production of metabolites. It's not only the individual metabolite that was increased, but also there was a big improvement on the yield. Yeah. So I think the yield went up to 50%. So we could say even 100%. See, so she doubled the yield from the molecule, which was really very good. So finally, I could conclude that natural products research could be amazing. Okay, in time of trying to find the right procedure to isolate these different compounds, but I think our metabolomic tools provided us a clear path where to go. Yeah, and I would say it's better than our room, the traffic signs. Maybe in Europe or here, I don't know, but this one is one example of a traffic sign down south. You go into a roundabout and you enter to several roundabouts. So there's a big possibility you take the wrong roundabout. Okay. Or you say good luck and then yes, which arrow you're going to follow. Keep right, but once the arrow going left. Okay, and beware of road surprises. That's very a typical scenario in natural products resource, but this is the scenario that we don't want to end up with. Yeah, is you don't want to be wasting resources. Yeah, you always want to be efficient. Yeah, if you end up with a scientist, the project is actually a failure. Okay. Finally, I would like to thank, of course, the universities we're working with. So Bangor University is actually the research with the two bells. FINEPS is Brazil, okay, to work with the Cerrado Flora. Okay, our funders, BBSRC, our Brazilian funders as well, CNTQ, CAPES, and FAPES. And of course, the people who work with us, Dr. Taufika, Dr. Sadata, right? And my recent PhD student is Nagu. That will be changed to doctor in 1 week. Okay. And of course, I would like to thank my Malaysian PhD students who work with me. Dr. Kamal Nurkhalida, who also became one of my postdocs later on, Dr. Maslan and Dr. Yusuf. Okay, so thank you for your kind attention.
Thank you very much, RuAngelie. Okay, all right. For very interesting, seminar lecture session, I think. So I open question to the floor and also to the online participants, especially the students. Please ask question if you have. Do you have any questions?
Yes.
This one way, it doesn't have the metabolites, but the Edrada-Ebel and that one. Do you think what kind of symbiosis that we have after they produce the novel compound to make it if they change each other or they put it together
Okay, so I think from the results that Elizabeth got, we think they're competing with each other. So for example, the combination of Alternaria and Fusarium, she will only get Alternaria. But there was actually a big improvement of the type of metabolites, right? So for example, there were new derivatives that were coming out, but it was mostly following the original Alternaria type of metabolite. Okay. So I think in the combination that we're doing, only one. Sorry. Okay. So do I need to repeat the answer? Okay. But they would not hear him. And they can they hear him? Yeah. Okay, I will repeat the question.
Yeah.
So the question was if the co-cultures, the individual fungi were playing a symbiosis or competing with each other. My answer is they are competing with each other because we could only see a dominance in terms of metabolite coming from one culture. Yeah, so for example, combination of Fusarium Alternaria, we could see the production, a higher production of metabolites coming from the alternariol type of metabolites, which is typically produced by Alternaria. The same thing with the two Fusarium species, yeah? One Fusarium, I think, took over the other because Elizabeth said, oh, look at this, I'm producing more of this noctiquinose that used to belong to these type of Fusarium species. Yeah. Mm-hmm. But those noctiquinose that she was able to later isolate, purify, yeah, I think four of them were new, and then three were already known as well. Yeah. More questions?
Is there any question? Ruan, I have one question.
Yeah, sure.
Regarding your fused metabolomic where you couple the LCMS metabolomics data with the NMR data. Because normally we use separately.
Yes.
Some of us working in the NMR metabolomics and some of us working on the LCMS or GCMS metabolomics. So in terms when you fuse the data, is there especially for the sample preparation in the NMR, for example, is there any guidelines what they should have in terms of preparation, the extract, for example, or is there any solvent that they need to use so that then they can?
Okay, so in terms of these data sets, we also use the same type of preparation when we don't use data sets. Yeah. So in our case for NMR, we're using DMSO titulated DMSO with 5 milligrams of extract fraction in 600 microliters. That's it. So we try to be consistent with our concentration, so because we don't want that to add as another variable or error when looking at the statistical analysis. When it comes to fractions, we use an extract, we use a concentration of 1 milligram per milliliter, yeah, with an injection volume of 10 microliters. Okay, I think a flow rate is 300 microliters. So it's still very tiny amount. But the concentration is important. You don't want to be having concentration as an additional point. Right? Regarding the solvent we use for mass spec, we use either a combination of methanol acetonitrile 1:1. We could also use methanol water if you're working with flavonoids. So students will usually use methanol water 1:1. Okay? Right. Or acetonitrile water 1:1. Just don't use any organic solvents with your mass spec. That will destroy your mass spec. Okay, and one.
Dr. Leong, one question.
One question. I saw standard finding anti-larval
Yes.
data, right? So wonder any further works on the compound that identified that related to anti-larval in the manner of how it actually functioning as an anti-larval that get into the cell that slow down the anti-larval process, or actually it's studying the larval itself. So it is
Right, okay. Thank you very much. So actually for the antibiofilm activity, we look at two processes. We have the P-biofilms, okay, which is inhibition of biofilm, which means the biofilm matrix will not form at all. Okay, and the second one is the so-called post-biofilm, which means the biofilm is already formed, but then the question is, do our compounds have the capability to disrupt or destroy the biofilm? So those are the only two mechanisms of action we're looking at in terms of biofilm. And then of course, the other test is the planktonic, wherein, okay, the biofilm is disrupted, then it kills the bacteria. So I think one important point is that one of the undergraduate projects was to do a literature survey on antibiofilm compounds supported in the literature versus the ones that are known to be antibacterial. And they found out that not all antibacterial compounds are actually antibiotics. Yeah. And Elizabeth had the same result as well in this. After my eyes are not flashing. And yeah, right. So not all the compounds that were found by Elizabeth as very good antibacterial activity gave us good antibiotics. It could also be vice versa. It will inhibit biofilm, but it will not necessarily kill the bacteria. Okay, so sometimes I think that's the reason why for multi-resistance you would use a combination of drugs rather than just one. Yeah, which is actually very good research.
And I have another question.
Yes.
I think what student experience here is maybe in terms of the annotation of the metabolites. They have quite something that they maybe have a issue to annotate. So is there any, I mean, suggestion or that you can tell them what are maybe the step that maybe they can go further if, for example, they're working in that specific species or genus or something like that.
All right, good. So regarding the application annotation, we don't really use MSMS at that stage. We only, because we already use NMR, so that will be our basis to say, okay, our replication result is actually correct because we could see these chemical shifts that pertain to this replicated compound, right? Now, that's one thing. The second is that we're using high resolution mass spectrometry, which means we are predicting the molecular formula at really, I think our error is down to 0.0, sorry. No, sorry. So 5 ppm is the borderline, while our error is down to 1, which is very good for an orbital. But I'm not saying I don't know what is with Brooker. What that is our experience with orbital. Now, when you get a lot of hits, we filter our hits using the family for the genes. Okay, so which means sometimes you will still get 100 hits, especially for smaller molecular weight compounds. So you need to filter it according to the family, the taxonomic identification, right? Another thing, the third one that I could add, anyone using Sirius here? Sirius.
Amalia?
Sirius. All right. I think some would pronounce it Sirius, yeah? But I think, yeah. I know who would be, so that's actually made by the European Enlightenment. They call it Sirius. Anyway. Right. No, that's not Enlightenment. It's a group from Chile. Okay. Sirius is very good. Okay, it's a freeware, so I'm not really advertising it too. Because it's not commercialized. So it's a freeware, so you could use it and it's very comprehensive. It covers everything. But a similar problem, it's difficult to filter because it covers something chem spider. And chem spider will have synthetic, microbes, plants, everything. Yeah, so we need to still filter it. Yeah, but it's a very good application to the science. Okay, but still the problem is filtering. So, yeah, we were discussing that when we met in Virginia. You should add that to your algorithm filtering, or maybe they would. So then, right. So if you want to look at it and spell SIRIUS. Yes.
I'm just actually really interested how we implement the visualization and the multivariate analysis with your condition and your environmental, like your polar temperature. Quite interesting how you did that analysis and you also like the concatenated between.
Yes.
I'm just wondering how we can always update our analysis of person. Because basically we are basically
Right.
infused with other kind of saturated and others.
Yes. I would say you always communicate with the developer. So this one, Simca is the developer is Symmetrics. And when we want to try something, I do communicate with them, say, okay, is this possible? When we started with Simca, Anita could really prove it, how basic Simca was in the beginning.
Yes, true.
They only they used to only have 2 colors, right?
Yeah,
Green and red.
true.
Right, and then said, oh, can you prove this? Yeah, you just talk to them. It said they're very open. All right, and then said, okay, we could prove this and that. No, When we started using Simca in 2010, it's very different from the Simca now. Yeah. So now we have Simca.
18.
18. I think we started with Simca 10. Oh, no, it's still called Syncopila. Syncopila. Yeah, Syncopila is the first. The same thing with MSETMAN. Well, we were lucky with MSETMAN because they were a partner in the European project. And they knew that, okay, our software or it's only working for biologicals, but not with natural products. So, and we're actually working at that time with between 100 to 500 samples. And MSETMAN cannot take it, but then they improve some of the parameters, they wrote parameters for us, they did a postdoc, and now the MSETMAN 3 is really far off from us. Now it's very quick. Yeah, try using MSETMAN 3. Don't use MSETMAN 4 yet. There's, I think, a lot of clicks. But they're still in the process. More questions?
Any question? Dr. Mike?
Yeah, online, right? You have questions?
No.
So I have a question.
Yeah.
So in this sharing of 10 years, may I know what the major challenge for the past 10 years and how you see the coming 10 years?
How do I see the coming 10 years? What's the challenge? Well, I think the main challenge is trying to innovate. Yeah, so you could see from the first year when we started together with Metabolomics, we have the basic applications and then strategy. So now the last year, it's changing it again. Okay, we apply Metabolomics and the elicitors, cultures. Yeah. So I think the current PhD students are actually also very curious, very innovative as well, and excited. As you will see, the development of software is also very quick. Yeah. So, well, I don't really find it as a challenge, more like completely tired about the work. So I think the main challenge is finding funding. I think that's key for everyone, right? Yeah. I think that's exciting.
How you see the next 10 years for you?
The next 10 years.
How old are you, will you?
Yes.
10 years, huh? The next
And
10
I
years.
think
How old are you now?
the next 10 years, then I would see maybe just like, oh, we action. Yeah, so, well, I had your own colleagues in the ICN, and they were just inspirational. And they saw, I think there's some of the speakers are already retired, still working as emeritus professors or research professors, where they're only part-time in the university. And I find it very inspirational because they can see how their network is coming out of their own lab, and that's how I would see it. For example, I would see Alida coming out with her own network as well, and whoever. That is how I see myself. So I think, yes, everyone knows, right? If you are an academic, that your success is, or the success of your students is your success. So that's how I was.
Question.
So the question is, I was wondering if in the course of assigned items, the Simca is able to identify isomers and assign IPs to identify to identify them before proceeding to the statistical part of the metabolism. If not, don't you think potentially something is okay. So Simca is only a multivariate analysis tool. It's not a replication tool. Simca will not be able to identify things for you. Yeah, it will tell you what to target, and you need to do that identification from a complementary replication tool. So that's the answer to that. Okay, Simca, it's a multivariate analysis tool. Second question. I am Kumo from Green, Indonesia. Hello. My question about the MSMS annotation. Do you have any suggestion for a simple but fruitful workflow to annotate MSMS spectra for untargeted metabolomics from medicinal plant samples? Ah, yes. Hi, Umo. You serious? Yeah, it's really good. Yeah, so my student, it has been a thing out there for only a year. And it's really great. So we've been trying it as well. Yeah. Mhm.
Alright. Okay, no more question. Is there any question? Oh yeah, Dr. Go has another question.
Comment about the patent.
The paper says.
Another topic, patent. Now, the patent, not
Patent.
then, would you take the pressure to patent finding?
Yes, okay. So regarding patent, I don't know what this is to itself, but we get a very good support from the university that's it for us. Yeah. So because Strathclyde is known for working already with the industry. So the industry partners are also very comfortable working with Strathclyde. But if you ask me the procedure, I don't have any idea how they do it.
But
Yeah.
you have to patent your finding before. Oh, oh,
Full 90, all right. Yes, we do. Well, it's not really, there's like a contract that the industrial partner will sign with the so it's always between university and industrial partner. If, for example, a university wants to patent something, and part of this was discovered by the university, then I think it's a certain percentage what the university gets and what the industry gets. Yeah. I don't know the exact, I just leave it to the university administrators.
they have been commercialized any of your.
None for my product, but I think in Strathclyde they have 2 products. Well, we have 2 products. I have 2 marine drug patents, but that is not the university. I was still in Germany.
Okay. So no more question, I think. But Ahmed, one more. Alright.
For the GHG, how much confidence would you like to separate that?
All right. When it's actually within the family, so for example, say, okay, one is for example, Edrada-Ebel, okay, and then we found or like anything for example, family. We have 100% confidence. Yeah, because we could see it isolated. But for example, if we have something like, okay, we have this metabolite, but it wasn't replicated to belong to another family or genus, or it's not even a microbial metabolite, it belongs to a plant, then we have less confidence. The confidence really goes down to less than 50%. Yeah, we need to isolate it or look at the NMR to MSMs. The problem is that when you have a relatively small compound, if you have a compound that is 200 daltons, then the MSMS data is not really so good.
It's not right.
Then if it's you could isolate it, then just isolate it and see what it does. You're welcome.
Any question? No, Dr. Kamal answer. I just call all the research fellow that work in metabolomics. All right. Okay. I think that's all. We have end to the question and answer sessions. So thank you very much, RuAngelie, again for your lecture. Okay. Before we end the session, I would like to invite our director from IN to give a token of appreciation. And maybe we can take group photo also. See here. Oh, online. Okay, online first. Okay, we take photo with the online participants. So for the online participant, could you please switch on your camera and get ready?
Okay. Dr. Halida, and can you stand in front of the camera to take picture with the online participant? Here, in front. Sekali. Sekali lah moderator. Online first. Online first, and then we take physical.
You just follow me. Come.
Okay. This one, this is the camera. Okay. Okay. 1, 2, 3, smile. Okay, another one. 1, 2, 3, smile. Okay, thank you, online participant.